The moment you swallow, you're kicking off a process that actually begins, what, roughly 4 weeks into your embryonic life?
That's right.
Welcome to the deep dive, where we take these incredibly complex biological sources and, well, we try to distill them into the high-yield knowledge you need to
Yeah.
understand the body's most intricate systems.
And today we are definitely undertaking one of the most structurally complex deep dives in developmental biology. It's the embryology of the digestive system.
This is a big one.
It is. We're moving past the basic layers today and diving into this really choreographed process of folding and rotation and molecular signaling,
Yeah.
all these changes that transform what is, just a simple endodermal sheet
Right, just a flat layer.
into the fully functional gastrointestinal tract.
And our source material here is, it's foundational. It focuses very intently on these critical events that really get going around the 4th week of development.
Hmm.
So our mission is to guide you, the learner, through all of it. The structure, the molecules, the clinical milestones of digestive system formation. We're basically tracing the path from a flat embryonic disc through the establishment of the foregut, midgut, and hindgut, and then ultimately to the very complex arrangement of the adult abdomen.
And I think the central theme here, the thing to really hold on to, is architectural coordination. It's all about coordination.
How so?
Well, we need to see how these initial positional signals, these chemical gradients, translate not only into cellular differentiation, what cell type to become,
Right.
but also into these huge, large-scale anatomical movements, where to go, how to twist and turn. And understanding that process is, it's the essential prerequisite for grasping the mechanisms behind the most common and sometimes life-threatening congenital abnormalities of the GI tract.
Okay, let's unpack this entire convoluted journey then, from a flat disc to a functional tube.
So we have to begin with the primary physical process that actually creates the gut tube in the first place.
And that's embryonic folding.
That's embryonic folding. It happens really rapidly around the 4th week, and it involves 2 simultaneous movements that basically convert that flat 3-layered disc into a cylinder.
Right, you've got the cephalocaudal, which is head-to-tail folding, and then the lateral, the side-to-side folding.
Exactly. And that whole movement, what it's really doing is it's grabbing that endodermal sheet, the one that formed back during gastrulation.
And pulling it inside.
Pouring it inside, yeah. So it gets incorporated, internalized right into the developing embryo. And the result is the primitive gut tube, just sort of suspended there within the central body cavity.
And a really crucial consequence of all that folding is the separation of the structures, right?
It's everything. The internalized portion forms what we call the intraembryonic primitive gut, while the folding itself, it sort of pinches off the connection points.
So things get left behind.
Things get left behind. 2 key structures remain outside the embryo proper. You've got the yolk sac and the allantois. The folding process basically creates a closed internal cavity for the gut to develop in.
Okay, so once that internalization and separation is complete, we can immediately start to define the 3 main segments based on their boundaries.
Yes. The cephalic folding, the head fold, creates the foregut. And it's a blind-ending tube at that cranial pole. It's sealed off by the oropharyngeal membrane.
A dead end, basically.
A temporary dead end. Then the caudal folding, the tail fold, it creates the hindgut, which is also a blind-ending tube, sealed off this time by the cloacal membrane.
So you have 2 closed ends.
Yeah.
And in the middle, we have the midgut.
The midgut. And this is the segment that remains temporarily connected to that big extraembryonic yolk sac.
Through that narrow stalk.
Through that narrow stalk, the vital line duct, or sometimes it's called the yolk stalk. So just like you said, 2 blind ends and 1 central connecting segment.
So while those are the 3 broad divisions, when we start talking about organ formation and especially blood supply, we usually use 4 more refined sections.
We do. And this detailed delineation is really crucial for, understanding the boundaries of adult pathology later on.
So the first one is the pharyngeal gut.
Right, the pharyngeal gut, or just the pharynx. And it extends from that oropharyngeal membrane down to the respiratory diverticulum.
Which is the lung bud.
Which is the lung bud. Now, for the purpose of GI tract development, the sources are pretty clear that this section is mostly responsible for forming structures of the head and neck, and we tend to kind of compartmentalize it from the rest of the tract we're talking about today.
Okay, so what's next?
Then we have what we call the remainder of the foregut. This is the segment just caudal to that pharyngeal tube, and it extends all the way down to where the liver outgrowth will be.
And its derivatives are What, the esophagus?
The distal esophagus, the stomach, and the superior or proximal part of the duodenum.
Got it. And the next major section is the midgut.
The midgut. This begins just caudal to that liver bud, and it forms the vast majority of the small intestine and part of the large intestine.
And it ends at a very, very precise landmark in the adult.
A very precise one. The junction of the right 2/3 and the left 1/3 of the transverse colon. And that boundary is so important because it dictates the switch from the superior mesenteric artery supply to the inferior mesenteric artery supply. It's a key vascular watershed.
Okay. And finally, the hindgut.
The hindgut. This section just picks up right from that junction, the left third of the transverse colon, and runs all the way down to the cloacal membrane.
And as you just mentioned, these three major segments, the remaining foregut, the midgut, and the hindgut, they're all supplied by their own distinct major arteries.
Absolutely. The celiac, the superior mesenteric, and the inferior mesenteric arteries, respectively. That vascular map is laid down very early.
So to really appreciate what forms, we need to quickly go back to the germ layer contributions. Because the gut tube might be one continuous structure, but it's really a collaboration between two different layers.
A very important collaboration. Each layer is responsible for totally different components of the wall.
Let's start with the inner lining, the endoderm.
Right. So the endoderm is the source of two primary components. First, the epithelial lining, the mucosa, of the entire digestive tract. That's the obvious one.
Right.
But second, and this is key, it's the source of the functional specialized cells, what we call the parenchyma of all the associated glands.
So we're talking about the hepatocytes in the liver.
Exactly. Hepatocytes in the liver, the exocrine and endocrine cells of the pancreas, all the cells that do the real work.
And then the surrounding supportive layer is the visceral mesoderm.
Or splanchnic mesoderm, yeah. This layer builds the structural framework. It forms the stroma, which is the connective tissue framework of those glands.
Oh, it holds everything together.
It holds everything together. And it also forms the musculaires, both circular and longitudinal, the general connective tissue, and all the peritoneal components of the gut wall.
So the endoderm provides the engine, but the mesoderm provides the housing and the power source.
That's a great way to put it.
Okay, so this is where it gets really interesting. How does a uniform, simple endodermal tube get its identity?
Right.
How does it know chemically that the segment next to the stomach has to become the duodenum and the segment after that should be the jejunum?
Right. The answer lies in the establishment of a molecular map. And it all starts with one crucial signaling molecule, the retinoic acid gradient.
So chemical signal.
Chemical signal. RA concentration acts as the primary master regulator, and it distributes positional information. This concentration gradient is very low or near absent in the most cranial region, the pharynx.
Okay.
And it steadily increases to its highest concentration in the caudal region, specifically the colon. This gradient literally defines the entire anterior-posterior axis of the developing tract.
And that RA gradient is what kicks off a cascade of different gene expression.
Exactly. Yeah. Different concentrations activate different transcription factors, and that establishes the initial patterning of the two.
The sources highlight four major ones. Right?
Yeah, they do. The four major transcription factors whose expression is graded along this axis are, first, SOX2. This is specified by low RA, and it dictates the formation of the esophagus and the stomach.
Okay.
Then PDX1, which specifies the duodenum. Then CDXC, which specifies the small intestine. And finally, CDXA, which is specified by the highest RA levels, and that governs the large intestine and rectum.
So that gives us the rough blueprint. But that initial geography, it needs to be stabilized.
It does. And that stabilization is provided by a really fundamental process in all of embryology, epithelial mesenchymal interaction.
A conversation between the two layers.
So reciprocal communication. It's a feedback loop between the endoderm and the surrounding visceral mesoderm. The process is actually initiated by the endoderm. Cells throughout the entire primitive gut tube start expressing the morphogen Sonic Hedgehog, or SHH.
And SHH acts as a signal to that outer layer, to the mesoderm.
Right. The SHH upregulates a very specific pattern of HOX genes in the adjacent visceral mesoderm. And these HOX genes are expressed in what's called a nested pattern. So, for example, you'll see HOX 9 through 13 expressed sequentially as you move down the tract. This nested code refines the regional identity that was first established by that initial RA gradient.
The sequence here seems critical.
It's everything. The endoderm signals SHH. The mesoderm receives that SHH and specifies itself using the HOX code. And here's the real core insight for the learner. Once the mesoderm has its HOX code, its final specific positional identity, the mesoderm then instructs the endoderm on the specific morphological details it needs to form the corresponding organ.
Wow. So it tells the endoderm what to become.
It tells the endoderm what shape to take, be it the cecum, the ascending colon, or the jejunum. The patterning is fully reciprocal. The endoderm starts the signal, but the mesoderm carries the blueprint for the final specific organ shape.
That structural refinement dictated by the mesoderm is really remarkable. It kind of explains why a defect in the surrounding connective tissue could lead to a defect in the specific epithelial lining, even if the endoderm's initial signal was totally correct.
Exactly. The layers are completely codependent.
All right, so transitioning from that molecular map to the gross structure, we have to talk about the structures that suspend and house the gut tube. The mesenteries and the peritoneum.
Yes.
The sources make a point to emphasize that while we use terms like intraperitoneal and retroperitoneal, the embryological origin of the mesentery is as a single, continuous, supportive sheet.
Precisely. Initially, the gut tube is broadly attached across the posterior abdominal wall, but by the 5th week, that attachment starts to narrow, and it suspends the caudal foregut, the midgut, and most of the hindgut via the dorsal mesentery.
And this narrowing creates a vital pathway. The dorsal mesentery, which runs continuously from the lower esophagus all the way down to the rectum, isn't just a physical tether.
No, it's much more.
It's the highway for all the essential supply lines. The entire arterial supply, the lymphatics, the nervous supply, it all has to travel through the leaves of this connective tissue sheet.
And its name changes regionally, which reflects the organ it's supporting, even though it is fundamentally one continuous thing. So you have the dorsal mesogastrium, the mesoduodenum, the mesentery proper for the small intestine, mesocolon, mesoappendix, mesosigmoid, and mesorectum. All parts of the same original structure.
Now, the pivotal moment comes when some of the organs lose their mobility.
Right. Organs like the ascending and descending colon, most of the duodenum, and the pancreas, they swing against and attach to the posterior body wall. This process effectively obliterates the mesentery in those regions,
Fixing the organ in place.
fixing it in a secondarily retroperitoneal position. They started out with a mesentery, but they lost it.
And this attachment creates a really clinically crucial structure called the Tolt fascia.
Yes. This is a fascial plane that forms between the visceral peritoneum of the attached organ and the parietal peritoneum that's lining the posterior body wall. For surgeons, understanding this vascular fascial plane is absolutely essential for safe dissection and mobilization of the colon and pancreas.
It lets them lift these organs away from the posterior structures without damaging blood vessels.
Without damaging the big vessels, exactly. It's a clean plane of dissection.
Okay, so if the dorsal mesentery runs the length of the tube, the ventral mesentery is much more restricted.
Highly restricted. It really only exists where the primitive gut meets the structures that are developing anteriorly. It's derived entirely from the mesenchyme of the septum transversum.
And that's that mesodermal plate that sits between the future thoracic and abdominal cavities.
That's the one. And the rapid growth of the liver is what partitions this ventral structure. As the hepatic cords start to penetrate the septum transversum, the ventral mesentery gets divided into 2 major components.
And those 2 components are
The lesser omentum, which is also called the ventral mesogastrium. It connects the stomach and the proximal duodenum to the liver. And then there's the falciform ligament, which connects the liver to the anterior abdominal wall.
Let's just nail down the definition of the covering membrane itself, the peritoneum.
Good idea. The peritoneum is a single, continuous serous membrane. The layer that's coating the organs is the visceral peritoneum.
And the layer lining the abdominal wall is the parietal peritoneum.
Right. And where the peritoneum folds to bridge an organ to the body wall or to another organ, those are called peritoneal reflections. These reflections, which are often called ligaments in gross anatomy, like the coronary reflections and triangular reflections around the liver, are key surgical dividers and landmarks.
Okay, so let's move specifically into the foregut. The development of the esophagus requires a really precise structural partitioning event to separate the digestive and respiratory systems.
It really does. And this partitioning begins around week 4 with the appearance of the respiratory diverticulum.
The lung bud.
The lung bud, yeah. It pops up on the ventral wall of the foregut.
And the subsequent division of that foregut tube is orchestrated by the growth of the tracheoesophageal septum.
Right. This septum grows caudally, and it cleanly divides the tube into a ventral part, which becomes the future trachea and lung buds, and a dorsal part, which becomes the future esophagus, a very clean split.
The esophagus is initially pretty short, but it has to lengthen dramatically. Why the rapid growth?
It has to lengthen because the heart and the lungs are rapidly descending into the chest cavity. They're growing and moving down. And if the esophagus fails to keep pace, if it doesn't lengthen sufficiently, it literally pulls the stomach up with it.
Resulting in a congenital hiatal hernia.
Exactly. Where part of the stomach ends up residing permanently in the thoracic cavity.
And what about the musculature? It's unique in its duality.
It is. The surrounding visceral mesenchyme forms the muscle coat. The upper two-thirds develops as striated muscle, which reflects its connection to the pharyngeal arch structures, and it's innervated by the vagus nerve, cranial nerve 10. But the lower one-third forms smooth muscle, which is typical of the rest of the gut tube, and it's innervated by the splanchnic plexus.
The clinical correlations here are, they're frequent and really important.
They are. The most common and serious anomaly is esophageal atresia and/or tracheoesophageal fistula, or TEF.
So what's the underlying mechanism?
It's a mechanical or a developmental error in that septum. It's either a spontaneous posterior deviation of the tracheoesophageal septum or some other factor that's pushing the dorsal wall of the foregut anteriorly. The split just doesn't happen correctly.
And the typical presentation, which I think occurs in something like 90% of cases, involves the proximal esophagus ending as a blind closed sac.
Yes, a blind pouch. While the distal esophageal segment actually connects to the trachea, usually just above its bifurcation.
And this specific anomaly gives us the classic mechanism for the prenatal finding of polyhydramnios.
Excess amniotic fluid. Exactly. Because the fetus is normally swallowing amniotic fluid, and the atresia prevents that fluid from passing into the intestinal tract for reabsorption. So the fluid just accumulates and accumulates in the amniotic sac.
We can also see just a simple narrowing, can't we?
We can. That's esophageal stenosis, and it often occurs in the lower third. It can result from incomplete recanalization, a process we'll see again in the duodenum where a solid tube has to reopen, or from some kind of vascular insult that compromises localized blood flow to the esophageal wall, causing scar tissue to form.
Okay, let's talk about the stomach. Its development is characterized by massive movement. It appears first around week 4 as just a simple fusiform dilation, a little swelling.
Right. But the final J shape and orientation are entirely dependent on 2 sequential rotations.
The first one is the longitudinal axis rotation.
A 90-degree clockwise spin when you're viewing it from the anterior.
And what are the results of that 90-degree turn?
Okay.
3, right.
There are 3 simultaneous results. First, the original left side of the stomach now faces anteriorly, and the original right side faces posteriorly.
Okay.
Second, the vagus nerves get dragged along for the ride. The left vagus nerve becomes the anterior vagal trunk, and the right vagus nerve becomes the posterior vagal trunk.
That's a classic anatomy question.
It is. And third, the original posterior wall grows significantly faster than the anterior wall, and that's what creates the massive greater curvature and the relatively static lesser curvature.
So once that longitudinal axis rotation is locked in, the stomach performs a second, more subtle rotation.
It does. The antero-posterior axis rotation. This is what aligns the organ diagonally. The pyloric, or caudal end of the stomach moves to the right and a little bit upward.
While the cardiac, or cephalic end, moves to the left and slightly downward.
Correct. And the result is the final adult position, with the axis running obliquely from above left to below right.
Now, these 2 rotations, especially the big longitudinal one, they don't just shape the stomach. They fundamentally reorganize the entire upper abdomen by pulling and reshaping the mesenteries attached to it.
They absolutely do. Initially, the stomach is centrally suspended by both the dorsal and the ventral mesogastrium, but that 90-degree clockwise rotation pulls the dorsal mesogastrium sharply to the left.
And that creates the omental bursa.
The omental bursa, or the lesser peritoneal sac, it's that critical space right behind the stomach. The rotation also pulls the ventral mesogastrium over to the right.
And around week 5, this rotating dorsal mesogastrium is the site where a major accessory organ forms.
The spleen. The spleen primordium emerges as a mesodermal proliferation between the two layers of the dorsal mesogastrium. And as that mesogastrium continues to lengthen and fold due to the stomach's rotation, the segment that connects the spleen to the midline swings left
And sticks to the back wall.
and permanently adheres to the posterior body wall via tolt fascia.
And this adherence is what defines the final peritoneal reflections of the spleen.
It does. We get two important connections from this. There's the lenarenal reflection, or ligament, which connects the spleen to the posterior body wall near the left kidney. And then there's the gastrolenal reflection connecting the spleen back to the stomach's greater curvature.
The pancreas positioning is also irrevocably changed by all this folding and attachment.
Indeed. While the head of the pancreas has its own story, the tail of the pancreas actually extends into the dorsal mesogastrium. And since that section of the mesogastrium becomes attached to the posterior body wall,
The tail of the pancreas gets fixed there too.
it's firmly fixed against that wall, making it a secondarily retroperitoneal structure, just like parts of the colon.
Okay, finally, the apron of the abdomen, the greater omentum.
The greater omentum results primarily from that differential growth and the anteroposterior rotation. The dorsal mesogastrium just bulges tremendously downward. It extends like a double-layered sac or an apron over the developing transverse colon and the small intestinal loops.
And eventually those layers fuse.
Eventually, the two layers of this apron fuse together to form the single sheet we see in the adult. And the posterior layer also fuses to the underlying mesentery of the transverse colon, creating that continuous appearance. It's a very complex bit of folding.
It really is. Okay, let's move on to the other derivatives of the foregut. The duodenum, liver, and pancreas.
The duodenum is interesting because it sits at a developmental crossroads. It's defined by its origin from two distinct gut segments.
Right, it's formed from the terminal foregut and the cephalic midgut, which means its junction point is precisely where that liver bud originated. And this duality defines its whole anatomy.
It does. As the stomach is performing its dramatic rotations, the duodenum is physically dragged along, forming its characteristic C-shaped loop and rotating to the right.
And that rotation, plus the rapid expansive growth of the head of the pancreas,
Causes most of the duodenum and the pancreas itself to become firmly fixed to the posterior body wall. Again, making them retroperitoneal.
But there's a small exception to that fixation rule.
There is the duodenal cap. It's a small proximal portion that often retains a small mesentery and remains unattached, which is why it's more mobile.
Let's talk about the critical process of recanalization. During the second month, the duodenal lumen goes through a temporary change that poses a risk for stenosis.
Right. During the 5th and 6th weeks, the epithelial cells in the duodenal walls proliferate so rapidly that the lumen becomes temporarily and completely obliterated. It becomes a solid tube for a short time.
And for the gut to function, it has to reopen.
It must undergo recanalization. Vacuoles form that coalesce to reopen the lumen, and failure of this process leads directly to duodenal atresia or stenosis.
And the definitive proof of its dual origin is its blood supply.
It's dual blood supply, yes. The proximal duodenum from the foregut is supplied by branches of the celiac artery. The distal duodenum from the midgut gets its blood from branches of the superior mesenteric artery. It is the only segment of the gut tube to get input from both of those major embryonic arteries.
Now, clinically, this segment is often implicated in pyloric stenosis.
A very common abnormality. This is where the muscular wall of the pylorus hypertrophies, the circular and sometimes the longitudinal muscle layers thicken severely.
And this creates an extreme narrowing of the pyloric lumen, which obstructs food passage.
It does. And the classic presentation is severe non-bilious projectile vomiting, which typically starts a few days after birth.
Now, the sources mention something interesting about the timing of this.
They do. While it's historically been considered a purely fetal developmental defect, there's data suggesting that while the predisposition may be fetal, the hypertrophy and the clinical presentation often manifest in the postnatal period.
So something after birth can trigger it.
Potentially. They specifically cite exposure to erythromycin treatment in newborns as substantially increasing the risk, which suggests a really complex interplay between development and the environment.
Fascinating. Let's move to the liver and gallbladder.
The liver, which really drives so much of the ventral mesenteric architecture, starts its development very early, like in the middle of the 3rd week.
It begins with the hepatic diverticulum, or the liver bud.
A rapid outgrowth of the endodermal epithelium at the distal end of the foregut. This diverticulum then rapidly penetrates that mesodermal plate we mentioned, the septum transversum.
And this invasion defines the germ layer contributions to the liver.
It does. The invading endoderm forms the hepatic cells, the parenchyma, and the lining of the biliary ducts. The mesoderm of the septum transversum forms all the supporting structures: the hematopoietic cells, the Kupffer cells, and the connective tissue, the stroma.
And the biliary system is a specific outgrowth of that connection to the duodenum.
Right. As the main hepatic diverticulum grows, the connection between it and the duodenum narrows to form the bile duct. Then a small, separate ventral outgrowth of the bile duct itself goes on to form the gallbladder and the cystic duct.
The relationship between the liver and the diaphragm also creates a really crucial anatomical landmark.
The bare area, yes. As the septum transversum thins out to form the ventral mesentery, the cranial surface of the liver, where it contacts the central diaphragm tendon, is never covered by peritoneum.
It's directly fused.
Directly fused. And the surrounding folds of peritoneum create the anterior and posterior coronary reflections, which then meet laterally to form the triangular reflections.
We also have that specialized free margin of the lesser omentum that relates to the liver.
We do. That free margin thickens to house the crucial vascular bundle known as the portal pedicle, or the portal triad.
The bile duct, the portal vein, and the hepatic artery.
All three. And this bundle also forms the roof of the epiploic foramen of Winslow, which is the necessary entryway connecting the lesser sac, the omental bursa, with the greater peritoneal sac.
Functionally, the liver is huge in the fetus.
It dominates the abdominal cavity early on, and that's because of its primary role in hematopoiesis, in making blood cells. The fetal liver is proportionally massive. At 10 weeks, its weight is a full 10% of the total body weight. Yeah, it's just filled with proliferating blood cells. This activity subsides during the final months of gestation, and it stabilizes at about 5% at birth. Bile formation starts later, around the 12th week, and as it enters the GI tract, it imparts that characteristic dark green color to the meconium.
Now, the molecular regulation of how the liver forms is one of the most, I think, intellectually satisfying mechanisms in the sources. It relies on inhibition, not direct activation. It's induction by removing repression.
It's the reverse of what you'd expect. The default state is that all of the foregut endoderm actually has liver potential.
So it all could become liver.
It all could, but this potential is actively repressed by inhibitory factors that are secreted by the notochord, the ectoderm, and the non-cardiac mesoderm.
So to get a liver, you have to selectively block those repressors right in the area where the liver is supposed to form.
Precisely. And the signal required for that comes from the adjacent cardiac mesoderm and nearby vascular endothelial cells. They secrete FGF2, fibroblast growth factor 2. FGF2 acts as the anti-inhibitor. It successfully blocks those inhibitory factors, which allows the gut endoderm to finally express its liver-specific genes.
And there's a secondary support signal that enhances the responsiveness.
There is. That's the role of BMPs, or bone morphogenetic proteins. They're secreted by the septum transversum. And BMPs enhance the competence of the prospective liver endoderm, basically allowing it to efficiently respond to that FGF2 signal. Then after that derepression, you get factors like HNF3 and HNF4, which regulate the final differentiation into hepatocyte and biliary cell lineages.
Okay, let's wrap up the foregut with the clinical correlates. Defects in the biliary tree are critical.
They are. Accessory hepatic ducts and duplication of the gallbladder are relatively common variations, and they're often asymptomatic unless they become involved in some disease process. The major serious defect, however, involves the biliary outflow.
That would be extrahepatic biliary atresia.
Correct. This occurs when the extrahepatic ducts fail to recanalize following their temporary solid phase. It's the same mechanism we just discussed in the duodenum.
But with much more severe consequence.
Much more. It occurs in about 1 in 15,000 births, and this blockage prevents bile from reaching the GI tract, which leads to serious jaundice and requires surgical intervention, often, ultimately, a liver transplant.